everyone. Thank you for joining us today for a webinar regarding the quality attributes for QC quality control release testing for mRNA as a drug substance. Next slide, please. So a quick housekeeping issues. The agenda would be welcome and logistic. Then Ashley gonna do an introduction to AMM and the AMM Science and Technology Committee webinar series. And then we're gonna have two speakers. We're gonna start with Diane McCarthy from USP, and then Erin Cowley from ReciBioPharm. And we'll end up with a roundtable discussion. Just to note, during the presentation, please, if you have any questions, put them in the box for questions, and we'll be more than happy to address those questions during the Q&A session and the roundtable discussion. Next slide. Again, the questions, there will be any Q&A box, and please put the questions in there and not in the chat box. And the webinar will be recorded and available at AMM website at in a few days.
Great.
Ashley?
Thank you. Next slide, please. So for those of us who may be joining the webinar series for the first time, the Alliance for mRNA Medicines is a group that's dedicated to advancing and advocating for mRNA and next generation encoding RNA therapeutics and vaccines. And our mission is really to propel the future of mRNA medicine and ultimately help to improve patients' lives and advance scientific knowledge. And we do this by convening industry leaders, innovators, scientists, and other key stakeholders. Next slide, please. AMM does a lot of different things, and part of our mission really is to educate and demystify the technology around mRNA. We engage with regulators, policymakers, and other stakeholders across North America, Europe, and the Asia Pacific region. We've been developing a really comprehensive source of mRNA sector data, and ultimately we like to convene the community to really amplify the work of scientists and organizations that are seeking to improve health through mRNA. Next slide. To that last point, we'd like to remind everyone to save the date for our second annual AMM Ascent Conference that will be taking place this year in Coronado Island, California, right outside of San Diego, from September 16th to 18th. All right, next slide. Again, along the lines of our mission to educate, we've been hosting this science and technology webinar series. Next slide, that's fine. And just as a heads up, we have our next webinar on August 26th, which will be talking about delivery strategies and processes for mRNA therapeutics. And we have a great lineup of speakers already lined up, including Dr. Peter Cullis from the University of British Columbia. So you can already go ahead and register for that webinar on our website. Next slide. We'll have a few other webinars to round out the year in October, November, and December. And those will have registration links live shortly as well. Next slide. For today's webinar, we have a great lineup of speakers. Colin Yamut, who is the co-chair of our Science and Technology Committee, will be the moderator of the roundtable that will occur after the presentations. And then we have Erin Cowley, who's the Chief Scientific Officer of ReciBioPharm, and Diane McCarthy, who's the Senior Scientific Director of Global Biologics at US Pharmacopeia. And with that, I'll turn it over to Diane.
Great. Thank you, Ashley, and thank you to AMM for inviting me to speak today. If you could advance probably 2 slides. Yeah, so what I really want to talk about today is around some of the work that USP is doing and how we're engaging with the broader mRNA community to build consensus and then to support development of standards and methods that can support quality of mRNA. So I'll give a little brief introduction to USP, talk about the consensus building, the analytics, and then a few comments on sort of where I think the field is going and how, how we can support that as well. So next slide. So just a brief introduction to USP. USP is a now over 200-year-old organization that has always been focused on quality of medicines. that started in 1820 with, really compounding in the early days of pharmaceutics. But, now we're looking at really how can we develop standards, tools, guidance that can help support quality of medicines, including mRNA. Our standards are used in over 150 countries and have legal standing in over, I think, 40 or maybe now 50 countries worldwide. Next slide, please. Within the biologics team, we support a number of different modalities, and you'll see some of these really aren't from a regulatory perspective, actually even biologics like antibiotics and peptides, although they do have more biological assays, and so that's why within USP they do fall within biologics. And mRNA is a really interesting one because when the pandemic hit and we were looking for expertise in mRNA, we found that this really bridged across vaccines as well as cell and gene therapy in terms of where that expertise was. And of course, it's also the areas where we see the most use of mRNA. We're quite interested in looking at some of the cross-cutting methods. And so when we look at how we can support quality, we're looking from really across the entire life cycle, from raw materials, obviously the particular PQAs, CQAs for the product, as well as things like impurities and excipients, and of course, microbiology testing that is really applicable across almost all therapeutics. And we're also really looking at how we can facilitate both adoption of enabling technologies. So, as many new technologies are coming on board, particularly for advanced therapies, including mRNAs, as well as enabling standardization or consensus building around new modalities. And so, I think that mRNA has been really a great journey for us, and an area where I think we can have a big impact. Next slide, please. Wanted to talk a little bit about how we work because it's quite unique. So we are really very stakeholder driven. And so we engage regularly with industry, with government across the globe, and with other subject matter experts to really try to understand where are the bottlenecks, where are the challenges, and what can a standard setting organization like USP do to help alleviate some of those bottlenecks. Some of the ways that we do that are through documentary standards, and I'll talk a lot today about general chapters, which I think is one of the most applicable areas for mRNA-based therapies. And then we also develop physical reference standards and reference materials that can help support quality testing. So really thinking about establishing your system suitability, or helping you do your quantitation for particular aspects. In addition to that, we are also very focused on education and training, and this was a big component of what we did really during the pandemic in terms of trying to educate people on mRNA as a new modality, big focus on the educating regulators around the globe. And then we do collaborate quite a bit with other organizations, and ultimately with the goal of enhancing quality throughout, again, the whole life cycle from R&D through manufacturing and distribution. Next slide, please. Want to comment on how we do what we do. So USP itself does not make the decisions on standards. We rely on our expert volunteers that serve on our expert committees to do that. We work in a 5-year cycle, and so we onboard experts from industry as well as other organizations onto our expert committees for a 5-year period. Our expert committees also will always include FDA staff and often regulators from other global regulatory agencies as well. So we're just starting our 2025-2030 cycle. We've just onboarded or just about to onboard our new volunteers. And within biologics, we will have this cycle 4 committees. So you see them sort of listed here and shown in the little circle. So most relevant are Bio3 and Bio4, which is the vaccines and the cell and gene therapy. And so some of the work with mRNA will really bridge those two committees. We also have committees focused on peptides, oligos, and complex carbohydrates, and then another committee focused on protein. And these are actually the people that will vote on the standards. So they review all of the documentary standards that come out, all of the new reference standards that come out, and they're the ones that really provide the guidance and ultimately the approval as we move standards forward. Next slide, please. And you could skip one more. So I wanted to talk a little bit about what we've done over the years to help start to build consensus around quality, attributes, and testing for mRNAs. And interestingly, we actually started looking at mRNA all the way back in like 2017, 2018. So long before the pandemic hit and many other people jumped on board with mRNA. We had our first roundtable on mRNA back in 2018. And while we discussed a lot of the same challenges that I think we see today, that there really wasn't yet, the field was really evolving very quickly. And so we didn't start working on standards quite at that point because you really have to let this field settle some before you're ready to start moving forward with standards. That being said, we were fairly well prepared when the pandemic hit. We had an understanding of mRNA and some of the bottlenecks. And we did a number of things to support COVID vaccines in particular. I won't talk about them today, but I'll talk more about really the analytics. So in 2021, we developed a vaccine toolkit, and I'll talk about this on the next slide. This was really focused on bringing together existing USP resources that could help people understand the data that they were getting, particularly global regulators, through the dossiers and the C of A's on the vaccines to really be able to interpret that information. We realized that that was a good first step, but it wasn't enough because there was a real need for methods and to start to build consensus on what were the CQAs that were important, what were the appropriate methods. And so in 2022, we released the first version of what we call the analytical procedures for mRNA vaccine quality, or I sometimes call it the mRNA draft guidelines. These were really a very early draft of some methods that could be used to assess CQAs, a list of CQAs. And the intent here was to be able to use this as a way to start to build consensus, get feedback from the community, the industry, and we were able to evolve those over the next couple of years. And we did a second version and then a third version in 2024. And I'll talk about how we're evolving those today as well. We also have done over that period of time a number of trainings, webinars, and attended a number of conferences. And we actually sponsored a couple of virtual conferences, the mRNA Open Forum in 2024 and the mRNA Summit last year. And in 2024, we also initiated a general chapter on best practices for mRNA, and I'll touch more on that. So this is really an evolving and very active area for USP. Next slide, please. So I mentioned on the last slide the toolkit, and I just wanted to show what this looked like. This was really, again, pulling together existing resources within USP that were applicable to mRNA-based vaccines. We actually did this for other vaccine modalities as well. So one of the things you see on the right-hand side in particular are the compendial tests so these are, things like appearance, pH, sterility, endotoxin that are going to be applicable to almost all drug products. And so these are already fully validated methods that people can use. And I want to comment maybe here on the numbering. So there are two types of general chapters within USP. Those chapters that have numbers under 1,000 typically have validated methods and often associated reference standards to help support those methods. And then those chapters that are numbered above 1,000 tend to be general guidance. So they'll provide you considerations, maybe different technologies that can be used. to measure similar attributes, and some of the sort of tips and tricks, but they don't typically have then a fully validated method. Next slide, please. So the toolkits were a good start, but our vaccine experts, both serving on our committees, as well as some experts that were part of a vaccine advisory group we had at that time, really felt that we needed more information. We needed actual methods that could be used to assess some of these quality attributes. And so we collected methods primarily from public sources for the first version. And then those draft guidelines were developed and reviewed by the vaccine experts serving on our committees. And as I mentioned in the last slide, we've been able to, through several public comment periods, we've addressed comments, we've gotten donations of a number of different alternative methods that now have been incorporated. And so that draft guideline has been expanded significantly from its first version that we released in 2022. We are now at the stage where we are evaluating many of the methods in those draft guidelines, working towards validating them so they could become official USP chapters, and developing the reference materials that would need to accompany those to help sort of support the method either through system suitability or as quantitative standards. And so the expectation is that, for many of these methods, we think that these can be sort of platform analytical methods that could be incorporated into USPNF. The other way that we're building consensus is through the development of a above 1,000 chapter, in this case, on best practices for mRNA-based products. And so this is really intended to cover everything from quality of raw material, even the design and manufacturing, through the analytical methods that are used, and things like stability and comparability. And so we convened an expert panel to work on this in 2024. And I'll maybe comment briefly on what an expert panel is. So I talked about the committees, but many times the committees come in with very broad expertise. On a vaccine committee, for example, you might have people with experience in different vaccine modalities. So if we really need deep expertise on a particular topic, which was the case here, we needed really deep expertise in mRNA, we will create an expert panel that works underneath that committee, but is the one that really drives the drafting of the chapter. The committee is still has the decision-making authority, but the panel does a lot of the work. And so here we brought together 14 experts from industry, as well as 2 government liaisons from regulatory agencies, and they have been working on drafting this chapter over the past year, and that work is continuing. Next slide, please. So I'll move on to the analytics. And while there are a number of steps, of course, in the manufacturing workflow, USP's primary focus is really on the analytical methods. And so we're really looking at this in 3 buckets. One is the raw materials, and we all know with the synthetic process used for mRNA, there's a lot of new raw materials that have not been used in other types of therapeutics, things like enzymes, nucleosides, capping reagents. And so controlling the quality of those raw materials coming into your process is critically important. So how can we develop standards, methods, et cetera, that can be used to support the raw materials? Also, many of those raw materials at the end of your process now become residual impurities. And so thinking about it from both sides, quality of the incoming and quality of the outgoing, to make sure that you've cleared actually all of those raw materials and reagents that were put in. And then, of course, in the middle, really trying to address the product quality attributes, trying to come to some consensus around what are appropriate test methods and what types of standards are needed to support those methods. Next slide, please. So I want to just comment on some of the basics that are relevant to biologics. On the left-hand side, these are resources that you may want to look at in terms of guidance on method verification, validation, and analytical procedures. One of the advantages of having a below 1,000 general chapter within USP is that those methods are already validated. And so you do not need to do your own validation. You just need to verify that the method performs appropriately with your particular product. And so that's one of the real advantages of moving some of these methods into a compendial chapter. One of the bottlenecks I often hear too with mRNA, one of the big areas of debate is around the bioassays. And so also quite relevant are the series of chapters on the right-hand side focused on bioassays, particularly 1032, 33, and 34, which have a lot of good information, everything from the design through the statistical analysis of the results of those bioassays. And then the compendial methods on the bottom, which I think I showed primarily, most of those on a previous slide. So next slide, please. I don't intend for you to be able to read this, but this is just an excerpt of the quality attributes for mRNA drug substance that was included in version 3 of the draft guidelines. And you'll see one of the things you'll notice is that we purposely tried to include multiple methods for the same the same quality attribute to accommodate the fact that different laboratories, particularly when you think about it on a global basis, will have access to different types of technologies, and so it's not a one size fits all. And I'm going to take a couple of these methods and really go into a little bit more detail around what we included, but also think about it from a physical standards perspective. What type of physical standards are needed to support those methods? Some of these may be things you need to develop in-house. Others might be things that we would be working on, or other standard setting organizations may be working on to support more platform analytical methods. And how are they actually used in the assay? So next slide, please. So I'll start here with mRNA content, which of course is a critical quality attribute because we are dosing based on the content or the concentration of the mRNA. The USP draft guidelines list a couple of different methods, both RT-PCR using both quantitative PCR and digital PCR. There's also a method in there for UV-based quantitation, which is probably more of a orthogonal method than a primary method. And there are a number of other methods that have been or could be used, including fluorescence spectroscopy and anion exchange chromatography. So this is actually this standard needs to support absolute quantitation of your mRNA, and so it's then considered to be a quantitative standard, and therefore it's very important to get the quantity of your reference standard that is used to develop that standard curve correct. And this is where in the USP process, we typically take our standards through multi-lab collaborative testing so that we can look at the performance in multiple labs and come and arrive at a quantitative value that is based on multiple labs, multiple independent assays. In this case, because you're using qPCR, you'd be developing primers probably to the UTRs. They're often company or product specific. And so this is not the type of standard that USP would probably develop. This is a standard you would need to develop in-house, but you'd really need to make sure that the quantity of that standard is very accurate. Next slide, please. Two other methods, so mRNA integrity and poly-A tail length. These are more amenable to the platform analytics, so to speak. For the poly-A tail length, for the mRNA integrity, we recommend a couple of different methods: fragment analyzer, CGE, and agarose gel electrophoresis. Again, not probably as commonly used. And there are other methods that are used here as well. This here, the method would be primarily used to establish system suitability. And typically for something like this, we would look at criteria for system suitability, like the similarity of the profile to the standard, to like a image of what this should look like, which would be on the certificates, as well as the resolution and the repeatability, often done with something like 6 replicates. This is a qualitative standard, although you would do relative quantitation of the peaks. But the absolute quantity of what's in there is not as critical here. And these can also be used to bridge to some of those other methods. Similarly, with poly A tail length, the primary methods are LCMS or IPRP, HPLC. This is really focused on sizing. So you would have a ladder of, and what we're developing now is a ladder of poly A tail lengths that then can be used for sizing. So again, this is not really quantitative, it's a qualitative standard, but you would use that to establish system suitability and then also to establish the length of your unknown sample poly A tails. Next slide, please. So lastly, I just wanted to comment on impurities and some of the major impurities and impact that they may have. So of course, we saw on the last slide, you can have truncated or fragmented RNA. You can also have extended transcripts. And that can actually impact then ultimately the translation and the amount of protein that you are really delivering to the patient as the active component. Double-stranded RNA, as well as residual DNA, are both considered to be risks for immunogenicity. And so it's important from a safety and a regulatory perspective to control those. And then some of the residuals like the enzymes, the reagents, some of the inefficiencies or byproducts from capping, for example, can affect both the product stability or the shelf life of the product. And so again, important to make sure that those have been adequately cleared. Next slide, please. So thinking about process-related impurities, you have things like the residual NTPs and capping agents, the enzymes and the solvents. There's always already some resources within USPNF on residual solvents. We're working on standards for residual NTPs and capping, and you can see the anion exchange method that we're using to do that on the right-hand side. So looking at both unmodified as well as the modified UTPs and the capping reagents as a way to quantitate the impurities. And again, here the quantity becomes really critical. Most of the time when you buy NTPs, it's a nominal concentration. It's 100 millimolar or 10 millimolar, but those numbers are probably not that exact. And in fact, if we look at nucleosides from different vendors, we do see that there are differences. And so we're working on, really getting a quantitative standard tested across multiple labs that can help with the absolute quantitation of these residuals. Next slide, please. And double-stranded RNA, as we mentioned, is also critically important. We've looked primarily at the immunoblot assays, which are not very technically advanced, but have been used consistently in filings, looking at both the absorbance and the chemiluminescent methods. While both work, the chemiluminescent method is significantly more sensitive. So this is the method that we're planning to move forward with and to develop the reference standards to support it. This is an area where we see a lot of development in new assays, new technologies to be able to detect double-stranded RNA. And so I think these materials will also be really useful in terms of bridging across some of those new methods. And we intend to actually test a number of those other kits that are out there as we develop these physical reference materials. Next slide, please. So lastly, I just wanted to comment briefly on where I think the field might be going and how we're thinking about it. And first I want to start with the platform analytics, which I've talked about this quite a bit, saying, there is opportunity for a lot of these attributes to develop platform analytics. And I just put in here the definition from ICH-Q2R2 and the new Q14, which is also incorporated into USP's Chapter 1220, where a platform analytical procedure has to be able to be used across multiple products without significant change. And so it requires that the molecules are sufficiently alike. So sometimes your platform analytics approach will work, and in other cases, there will be some challenges if you have a much longer mRNA, for example, or a different LNP composition. One of the big things I think that comes out of Q14 and the updated Q2R2 is that we have now more ability to leverage prior knowledge, and that's also allowing us to be more efficient because we can do a more abbreviated development relying on some of that prior knowledge. The other component here is really taking advantage of the QBD principles and the analytical life cycle management. And let me actually, if you go to the next slide, it has a little bit more detail. Oh, sorry. Oh, we'll go to that and I'll come back. No, I'm sorry, go to the next slide. Yeah. So for platform analytics, I think that platform analytics will be applicable across a number of different things: impurities, raw materials. Many of your sizing standards should be able to be applicable, but there are some exceptions. So some of the larger mRNAs, including some of the self-amplifying mRNAs, are really beyond the size range that most of the methods that are being used today can really accommodate. So that's an area where a platform method that's designed for an mRNA that's 3,000 or 4,000 nucleotides may not work well for someone that's 9,000 nucleotides, like a SAM mRNA. Also, potency, of course, is something that probably will not be platformable. And then any of the major changes you might make, changes to UTRs, may impact any of the qPCR, new LNP components, et cetera. So if you go to the next slide, coming back then to that life cycle management. Oh, one more slide, sorry. Sorry, go back one. The other thing I wanted to comment on here is that there are so many methods being developed for mRNA-based therapies. And so this is a very active field. We're going to see a lot of new technologies coming on board. And so reference standards really can play a key role here in bridging to new analytical methods. And so having these physical materials available as we get those through development will really help in adoption of some of these new advanced methods as well. And the next slide, I think we'll talk about the life cycle management. Yeah, I wanted to really highlight this, not to go into detail, but a lot of the information around platform methods and how to use the QBD approach to develop your quality target product profile and then your analytical target profile are in Chapter 10, 1220 on analytical procedure life cycle, which was published a couple of years ago. I did want to mention, because it is currently open for public comment, that there is a new chapter that really focuses on stage 3 that's highlighted there on the ongoing procedure performance verification. So this is really expanding that information that was in 1220 on stage 3 into its own chapter. This was developed by 2 of our expert committees, the Statistics Committee and the Measurement and Data Quality Expert Committee. And it is open for public comment through September 30th of this year. So I really encourage you, if this is an area of interest, to take a look at this. Pharmacopeia Forum is free. You do not have to have a subscription to USPNF. So anybody can go in there and review and then provide comments. And those comments will all be addressed by the expert committee once those come in before they release the next version. Next slide. So just to wrap up, a perspective on sort of opportunities for standards and things to think about. Clearly, we need an expanded portfolio of standards to help support quality of mRNA across the entire product lifecycle. From a documentary standards perspective, we're working on both the best practices for mRNA products, as well as validating some of the methods from the analytical draft guidelines. And I'd like to comment here too that we are still very much interested and open to accepting additional methods that we could move forward. I know as technologies are advancing, there may be new methods that would be worth including as we move forward with these documentary standards as well. In terms of reference materials, we're looking across, again, the entire life cycle, trying to develop materials that'll help and support the consistency and the establishment of system suitability for the analytical methods, as well as really looking again at how we can support some of the new analytical methods. And then finally, we continue similar to AMM to really try to support training and education on mRNA, as well as sort of analytical transfer and life cycle management overall. And then the last slide is just to highlight some areas for opportunities for collaboration. We certainly encourage you to join some of our events. You heard about the expert panel on mRNA, so we don't have an open recruitment right now, but we will have additional conferences, workshops, etc., that we will be sponsoring throughout the year. If there are methods that you're interested in seeing incorporated into USP standards, we're certainly always open to you sharing those methods or materials with us that can help support standard development. And for many of these, we will also be doing collaborative testing, where we do that multi-lab testing to establish the quality attributes or the quantity, et cetera, of the standards. And so where we are doing collaborative testing and we're looking for labs, those notices will be posted on our website. And then finally, encourage you to take a look at the chapters and the things that come out in Pharmacopeia Forum. Right now, 1221 is out. But when the mRNA expert panel is done with their chapter, that will also be coming through Pharmacopeia Forum, and encourage you to comment on that. With that, thank you for your attention, and pass it back to the AMM team.
Thank you. Aaron, you're up next.
Yeah, it's actually a pleasure to be here and actually talk to everyone here today. So what I'm going to focus on is actually the analytics and kind of the future of it's actually PAT for RNA. It's actually next slide. So it's actually what I'll talk about here in the next extra 25 minutes or so is kind of give you a little bit of background behind actually ReciBioPharm. It's actually the analytics that we're actually deploying. Kind of the vision for actually meeting the industry's needs, and actually what we're doing about that. And then finally, to actually conclude, it's actually what we talked about and have actually built here. I think it's important to just point out that we actually do work with actually a number of institutes on what we're actually developing. So we have actually a very large grant from the FDA, the Gates Foundation, as well as actually MIT. That we're working very closely with. Next slide. So a little bit about it's actually ReciBioPharm and actually ReciBioPharm. So actually ReciBioPharm is actually it's actually CDMO globally. We have about actually 17 sites around the world. It's actually 5,000 employees and et cetera. So it's actually a relatively large actually CDMO. And it's actually ReciBioPharm, which is actually the Biologics actually division of ReciBioPharm. It's actually next slide. So this is kind of showing actually the three different business units of ReciBioPharm. So ReciBioPharm, which is the biologics side, it's actually aseptic fill finish and actually OSD. So those are kind of the three main, it's actually platforms or the three main, it's actually technologies and spaces that actually ReciBioPharm plays in. It's actually next slide. So in 2022, 2023, it's actually ReciBioPharm actually acquired 3 sites. It's actually one site, it's actually Watertown, Massachusetts, where I'm actually based out of. It's actually second site is in Portugal, and then there was a third site in Germany. So these extra 3 sites make up actually the biologics actually division of actually ReciPharm. And I'm actually primarily actually out of the US site. And so I'm just kind of looking at what we focus on. So that's actually nucleic acids, it's actually viral vectors, it's actually microbiome. So we're really focusing, it's actually on actually the ATMP space. And it's actually very similar to actually the Portugal, in actually the offering that we provide there as well. Next slide. It's actually across actually the 3 sites of the biologics division. You can kind of see the wealth of experience so that we're working with. So actually the number of actually customers that are actually that are ongoing or actually in the clinic. It's actually how many products that we're manufacturing for our partners. So it's actually the commercial programs, for example. And this is just it's actually over the past actually 3 years is kind of the metrics that this is actually highlighting here. Actually, next slide. Now, when it kind of comes actually specific to actual RNA, in actually the Watertown site, so this is kind of, it's actually specifics around that. It's actually the expertise is actually it's actually gene editing, it's actually rare disease, it's actually oncology. We have experience with messenger RNA, circular RNA, it's actually self-amplifying, it's actually guide RNA, silencing RNA. As well as actually multiple formulations at this point. It's actually very thankful to work with actually a number of customers to actually manufacture their materials, that actually have resulted into the clinic. And we have actually a number of actual batches that are scheduled. It's actually over the next coming months. It's actually next slide. All right, it's actually next slide. So in terms of actually the current, it's actually analytical, it's actually offering that we have, being a CDMO, is, we have actually the platform methods, which is essentially actually the plug and play methods, so that we can actually use across multiple programs. So it's actually the compendial methods, that are actually already established. It's just kind of like this is actually Diane mentioned, in actually the previous talk. And then it's actually the customer, this is actually specific methods, that it's actually based upon it's actually construct, et cetera, that's actually being developed. Next slide. So being actually a CDMO, it's actually very important to basically it's actually onboard as many of actually the analytics as actually possible. Because it's actually oftentimes if you're having to go through actually a third-party supplier, it's actually offering from a service perspective, so it just makes things a little bit more, it's actually complex and you're actually at the mercy of actually someone else's timing. To be able to kind of actually spearhead actually your assays that you have ongoing. But this is kind of actually a sample list of actually the assays so that we actually have available here at actually ReciBioPharm. And we basically support, so it's actually ICP testing, it's actually release testing, it's actually stability testing, it's actually raw materials and etc. It's actually next slide. So this is kind of it's actually more of actually the all-inclusive in terms of the assays and actually some of the methods that we're actually using or actually deploying for a lot of actually these assays. So I know there's a lot going on here, so I won't spend actually too much time, but so this is just kind of, it's actually a snippet of actually the technologies and methods. Actually, next slide. Now, so we actually support actually the full actual product, it's actually a production cycle. Basically it's going from actually the information transfer, being a CDMO, so it's actually onboarding a program from a client. It's actually bringing in actually the methods that they've actually developed and etc. So the second fee, it's actually a layer of that. So it's actually analytical. It's actually development. And that's actually what we have actually a number of scientists that are just focused on actually it's actually on that actual perspective. It's when we actually onboard, it's actually a program and actually methodologies. And then there's actually the qualification of the method. And then finally, it's actually release testing, and actually, this is actually the stability testing. So that's that's kind of the pathway that we actually take with, it's actually the clients that we actually onboard. Next slide. So this slide here is kind of showing, it's actually the release assays for, it's actually purified plasmid. So you're seeing actually most of actually the panel or actually platform methods as well as actually platform, it's actually qualified methods, which actually typically, so it's actually the impurities are the ones that get actually qualified. And so the stage of this is based upon, it's actually the early stage, of actually phase 1, phase 2. It's actually, so actually obviously it's actually as you get to phase 3 and actually commercialization, so pretty much all your methods get qualified and validated. But so this is just kind of showing you that this is actually a snippet for actually most of the clients right now. Next slide. This is actually, again, kind of showing actually the release panel for, it's actually drug substance for actual RNA. Again, you see actually most actual methods are platform. It's actually typically the potency, it's actually method is something that we're actually onboarding, and that's actually specific for actually the customer. Next slide. So this one here is actually the drug product side, kind of the release panel. Again, you see it's actually most actually methods are platform methods, but again, it's actually the lipid content, and this is really, it's actually driven based upon the formulation that is actually tech transferred to us. So that's actually, it's actually custom methodology. So it's actually the container closure, it's actually whether vials and syringes, it's actually whatever is kind of selected for the customer product. And, so the third thing is actually the potency. So that's actually the customer specific. Next slide. But so what I will go into next is kind of, so the vision that we have to kind of take actually the analytics, beyond what is just actually shown there. We have actually a pretty large grant from the FDA, like I mentioned. So it's actually $82 million. And we started this about 2.5 years ago. And we actually went out and actually did a VOC to actually see kind of what was actually most interested the people in actually the RNA field. And it was great to basically see PAT. So it's actually process analytical technologies, and actually using actually machine learning and actually AI are two of the big things that was actually most exciting about the field. So that was actually very exciting. So the middle part was just, actually the length of time to kind of do a program, to actually start from a construct and be able to get actually clinical trial material can be quite lengthy. It can take it's actually up to a year to be able to do that. And so how can you actually condense that? And then the third element is to the right. We all have actually a lot of, it's actually analytical testing, but typically most facilities have, it's actually QC laboratories. And so it's actually oftentimes in actually manufacturing, you're taking samples and you have to go to the QC. So you have to get it tested before you can actually push the product along. It's actually the manufacturing train. So it's actually the inefficiencies in actually manufacturing was very clear. And this is not just actually specific for RNA, but it's actually ATMPs as a whole. And so there's a lot here to kind of solve. Next slide. So we actually took this this money, and so we're building actually a number of elements, actually a platform. But what I want to just do is actually focus on the two in the middle. So what we actually decided to do is to use PAT as much as possible. It's actually throughout actually the manufacturing, so that we could fully actually integrate the production line without having to take samples to the QC. That has actually been the overall goal. And the only way you can do that is actually analytics. So you have to put eyes that's actually on your product at all times, and actually have to know how it actually impacts. It's actually the impurities, it's actually so the potency and etc. So this is a huge actually initiative to be able to make this a reality. It's actually next slide. So what we actually end up developing is what we call, it's actually a PAT skid. I should like to say it's actually a QC on wheels, where we basically have taken actually a single skid. It's actually there to the right, and we've actually incorporated, multiple, it's actually analytical technologies on it. So like this one actually in particular it's actually UV vis, it has actually mid IR, it's actually near IR, Raman, mols, it's actually DLS. So that's all on this actually single skid. And it's actually able to operate off of one software. So there's actually one software that runs all of actually those actual methods. It's actually all in one. And it's actually, as you can see, there's kind of multiple layers to actually the skid. So we can swap out, it's actually other technologies, and I'll actually kind of explain why here shortly. Actually, next slide. So this is kind of actually, it's actually starting to show the software that we've actually had to build with this. Because so what we've actually are building is a software program that we're able to basically build the models of, it's actually the inline analytics versus the offline analytics. And this is how we're able to start to actually show this. And actually those models is what gets plugged into the software. And so when it measures through this PAT skid, you basically get, it's actually check mark, check mark, check mark. So if it passes all of actually the CQA testing. But this is actually showing kind of it's actually the imagery of actually the IVT, where you're seeing actually the emergence of actually the RNA construct, and actually each one of actually the NTPs. You're seeing those actually decline as a function of time. So it's really putting eyes on the process, and so you know when to actually push the manufacturing along, because you can actually see and measure these actual elements in real time. And that's actually very important. It's actually next slide. But, ultimately, so this is actually it's actually technology that we're actually incorporating into manufacturing. So I like to consider the PAT skid being almost like, it's actually the Intel chip to be able to build actually the computer. So it's actually the backbone that goes along actually your full manufacturing train. So what you're essentially doing is putting a PAT skid. It's actually in between each different unit up. It's actually measuring the CQAs and then pushing it forward, and actually providing actually the control mechanisms. So the PAT skid is actually, as well as the software, is actually able to reach into the process unit ops and be able to actually extract data such as pH and temperature, and so it's actually stir rate and etc. And it's actually able to reach back in and actually control those elements as well through actually the PAT. It's actually software. So it's a really kind of a cool way to be able to actually mobilize your PAT and to actually use it. It's actually not only in PD, but in manufacturing, and also start to build in the controls for your manufacturing process. Next slide. So this is kind of actually the CP2 production system that we basically built for actual RNA, and you're seeing kind of these PAT skids built throughout, and it's being ran by a central, this is actually a control tower, it's actually a tower, is actually able to control this entire production train. So this is what's going to be fully on site by the actual end of August. So you're seeing actually a number of the skids are already here because you see the pictures. As well as the PAT skids, and we're going to be doing actually the commissioning this. It's actually throughout the actual remainder of the year, and then actually basically hitting go to start to actually showcase this in reality. It's actually next slide. This is kind of showing, it's actually the CQA coverage. So in green right now, so what we're able to measure with, it's actually inline analytics, is we're able to cover about 56, it's actually percent of all the CQAs right now using actually that PAT skid. And so what we're actually, so where we're planning to be by the end of the year is being able to cover, so that's actually 92%. So the ones in blue right there is actually what we're focused in right now, but the green ones are pretty much already validated, and so the blue are the ones that we're actually focused on right now and actually getting those, it's actually into play. Now, so we're not going to be at actually 100% coverage because there's still actually some technologies that actually need to be it's actually further developed to be able to get towards actually 100% coverage. But I think it's kind of safe to argue that we probably don't need to be it's actually 100% coverage. It's actually nice to have, essentially. But it's real exciting to be able to see and get to this point. And it's actually not only it's actually CQAs and what we're measuring based upon, this is actually a qualitative measurement. It's actually measuring down, to actually the quantitative is actually how these methods have been developed. Actually, next slide. So this is kind of showing, it's actually like a snippet of it's actually the traditional, it's actually offline, methods. So it's actually the RSDs, that are actually, it's actually for those methods. It's actually versus some of the current, this is actually inline actually methods that, so we've actually already deployed. So I'm not showing all of it here, but I'm just actually showing what I feel this is actually comfortable. It's actually showing in this presentation today. But you're seeing with actually the inline analytics, we're getting it's actually relative to the same. So it's actually so it's actually, if not better than it's actually the offline analytics, which is actually the gold standard. So it's actually real exciting to be able to show it's actually where the team has been able to get by incorporating actually PAT technology throughout the it's actually production train. Next slide. Now, so the benefits of actually being able to actually deploy PAT are kind of threefold. So kind of the top part of that is it's actually the traditional batch manufacturing where you have a QC laboratory that's gating. Typically if that's occurring, it takes roughly it's actually 21 days from actually IVT to fill finish. So the middle part is the current platform in the PD laboratory. So what we were able to do by incorporating PAT is being able to condense that. It's actually condense that down to 5 days. And the reason it's only 5 days is because we've been actually very busy building the models and it's actually identifying the PAT technologies that's actually needed. So it's actually doing the validation and et cetera, which takes actually more time so than what people kind of realize. And then finally, so it's actually the GMP system there at the bottom. So it's actually by being able to have it's actually these PAT, it's actually methods qualified, more of actually the CQAs incorporated. So what we're hoping to basically be is being able to condense that down to less than a day. And so that's where we're actually aiming to be. Now, so are we going to be there in 6 months? Probably not. If so, it may take another year. So it's actually 2 years to be able to get there. But it's actually in any event, it's actually going from 21 days, to 5 days. It's actually down to it's actually X days is actually a huge improvement. It's actually over the current. It's actually timeline. It's actually next slide. And so what we've been able to already show so the savings of both costs and time by actually deploying PAT. So again, so we're covering it's actually 56% of the CQAs today, is we're able to show roughly about actually 50% savings in both costs as well as actually labor hours by being able to actually deploy PAT. And so we're hoping to be closer to it's actually 80% by actually the end of the year. So it's actually checked back in by the end of the year, and so hopefully we're closer to it's actually the 80% target there, to kind of really make this a reality. So there's a huge savings that's actually starting to occur by it's actually adopting PAT. It's actually next slide. And it's kind of the ultimate goal is by actually deploying PAT throughout your entire facility now. It's actually in process development, it's actually analytical development, manufacturing, and et cetera. So we're able to kind of shift the entire timeline of, so it's actually being able to actually onboard a construct and it's actually making actually clinical trial material. It actually typically takes about 12 months. Well, it's actually kind of based upon it's actually market conditions and et cetera. So now we've been able to shift that down to about less than 4 months, by being able to put it's actually analytics everywhere and really be able to maximize the amount of data and use that data to really kind of drive actually the decisions in PD, it's actually analytical development as well as manufacturing. Next slide. So this slide is just actually already starting to show kind of actually the proof in the pudding that basically it's actually using the batch manufacturing versus it's actually integrated manufacturing of PAT. So we're able to get about it's actually the same quality of material it's actually already. So that's actually very exciting to be able to see that, and that should only improve. It's actually as actually the models improve. It's actually as pretty much actually everything improves with actually the integrated approach that we've developed here. Actually, next slide. So kind of the framework, so this is something that we're actually looking for people to start, doing evaluations. So it's actually beta testing actually towards the end of the year, but we do plan to actually start this. It's actually as actually an offering. It's actually to anyone who would want to use this and actually deploy this. It's actually in their laboratories, PD laboratories, manufacturing, it's actually et cetera. So this is something we're very exciting to be able to launch. Section next slide. How we actually see this being kind of adopted, the PAT skid is, it's probably for the first couple of years, it's really probably see it, it's actually beta testing in actually PD labs, it's actually 80 labs, really starting to build up actually the data that's needed for all the model development, for actually certain, it's actually constructs and et cetera. And then, being able to actually, actually progress the actual manufacturing floor more for, so it's actually IPC testing. It's actually on the floor. And so you're kind of actually alleviating actually the bottleneck from actually the QC laboratory. And ultimately, where I would love to actually see this go, maybe it's actually 10 years down the road, is truly, so it's actually mobilizing QC. So essentially how these PAT skids are built today is that we have actually different platforms or so that you can pull out and add in. So not only are we doing so it's actually inline analytics, but we're also building in so it's actually at-line it's actually analytics as well, such as it's actually flow cytometry, it's actually CE and et cetera, because so you can't solve everything with actually so it's actually inline analytics today. So you need to be able to have both actually inline as well as actually at line to truly get towards it's actually QC on wheels. So actually next slide. Finally, to actually conclude this is, it's actually ReciBioPharm is actually a well-established actually CDMO in actually the RNA sector as well as actually ATMPs. It's actually analytics is kind of the core foundation of actually the service offering that we provide. So we really feel that we're poised to actually radically kind of actually disrupt how these products get actually developed, it's actually manufactured and released by actually deploying these PAT skids. And it's actually it's actually test case is actually RNA, but the PAT skids are actually designed to actually be so it's actually agnostic, for actual modality, so you can, so actually a lot of the same, so actually analytics for RNA is actually very similar to MABs and actually other things. And so you can be able to swap things in and out. So we've already been able to show that we can reduce, the actual timelines by actually at least actually 50% from a PD, it's actually manufacturing and actual release, it's actually standpoint. And it's actually the regulatory, it's actually large pharma are actually very excited about the approach that we're taking here. So we're very, it's actually fortunate here. And it's actually next slide. I think that actually concludes my presentation for today, but it's actually, thank you very much.
Thank you, everyone. Thanks, Aaron and Diane. Great talk. Appreciate it. Just a reminder to the audience, if you have any questions, please put them in the Q&A box and we'll be more than happy to address them. I do have a couple of questions, and those are open to both. I see, Aaron, you have something called platform and unqualified platform assays.
Mhm.
Before I jump into that question, is when you defining a platform method, how are you defining that? Are you saying that this method is tied up to a standardized and locked-in process, or that method is available to be changed? You know, if there's any change in the process, that platform method will support that.
It's So it's kind of actually the combination of the both, to be honest with you, but it's really kind of a platform that's been developed for, it's actually multiple constructs. Now, it's actually when we do bring in a construct, we are kind of evaluating it against, all those controls, to be honest with you. And so there may actually, it's actually have to be tweaked, but so there's no actual major tweak, if that makes sense. So,
So,
but
so you'd have, so you'd have the T7, the tail, the cap, the UTR all fixed. It's
right.
just a minor thing that, okay. So, so that leads me to the platform versus qualified platform. So when you say it's a qualified platform assay, does that mean the method, like residual anti double-stranded RNA, it's regardless, you go through that my method and I'm going to be able to use that method for that. But when you say unqualified platform method, that more of like it's a generic method. If there's certain changes, we have to look into the method.
Yeah, it's kind of the qualified is kind of restricted more towards it's actually the safety based kind of actually assays. It's actually the impurity profile. So it goes through a little bit more infectious scrutiny, in terms of it's actually the qualification, right? It's actually of the method. It's actually before it goes into validation and et cetera. So, and that's really kind of how we define that for kind of that early stage, it's actually preclinical phase 1, phase 2, and then really start to go into validation. It's actually so the product, this is actually the life cycle, kind of continues actually down the clinical program.
Okay. Now, Diane, when you talk about analytical platform, we're talking about strictly analytical platform in the sense of what maps went through, not necessarily a process platform.
Correct. When I'm talking about platform, I'm talking about the analytical methods, not about the manufacturing. I think
Okay.
that is a different animal, right? But both can be platformed, so to speak.
No, no, I mean, I'm asking this question, and this is an open-ended question, for you guys to elaborate as much as you want on that, is, platform is used very loosely. And I just want to make sure, the audience or, who are listening to that, they do understand there are certain limitation to the definition of platform. It's not just run with it. So, the one of the things that, doing this for so long, there's always hurdles of transferring from a characterization into a QC release assay. So for both of you, what hurdles that you are encountering during that, for mRNA? However, want to go first,
Maybe I can comment quickly. I mean, I think obviously there is room for both. And I think as we are thinking about the methods that we would move into a compendial chapter, we are generally focused on those that can be used in the QC component, so they can be used at that for release testing. That being said, I think as we develop reference standards, we also recognize that there's a lot of other technologies that people are using. And so, one of the things we do quite routinely, and we've done it with a lot of our gene therapy standards that we've been working on over the last couple of years as well, is to take, the physical materials still should be a bridge between methods. We know the quantity, we know the purity, we know the key attributes of those physical reference materials. And so even if we don't necessarily have a method that's fully validated with a new technology, we can certainly, we do typically include, or we will add on even, after the initial release of a reference standard, what the data should look like with new analytical platforms so that you have that option. And then those might be used today in characterization, but maybe, over time, those will be implemented for release assays as well.
Yeah, I mean, I would say from our side, I think we've had a very unique situation here where, we're doing a lot of, so it's actually the tech scouting it's actually analytics that are actually there today versus, being used actually somewhere else, I mean, it's actually airline industry or whatever. And it really comes down to kind of, being able to actually reduce it. It's actually the variability in the assay, right? And by really being able to target such a specific, so it's actually NMR is a prime example. So actually NMR is a very powerful instrument that you can get great separation. Of actually a lot of, it's actually small molecules. So you don't have a lot, of actual overlap. And we've screened actually multiple constructs and methods, and we're still getting actually very defined peaks and et cetera. So that's why it's actually the percent error is so low. And that's actually very critical from actually the control perspective in manufacturing, but also just from it's actually release perspective to be able to really tighten these down, if that makes sense.
So, this leads to another question of there's a lot of talk been how do you correlate, your physical attribute tests, which is, lots of the stuff being done, with this concept you see on wheels or, the current methods that are available to correlate that to a functional assay. Because at the end, mRNA is really unique in terms of, it does something to produce something. So you really have to look at the functional. So how do you guys see that futuristic, you know, a, a true functional assay could replace quite a few of the physical assays? Uh, what's your thought on that?
So that's an interesting. So that's the one that was still white on the screen. So there's a lot of thoughts about that, to be honest with you, but it's actually a little bit probably premature for me to speak with it. But because a lot of actually the cell-based assays and et cetera takes time. And so yeah, it is tricky, to be honest. I don't have an answer for you today.
Okay.
Yes, I know my team is looking at it, but yeah.
Okay. Diane, do you see USP moving forward with some certain standards to help with the potency, with those functionality assays, uh, it, to transition to a good QC assay, for release in terms of functionality?
Yeah, I think it's a challenging one. I know, potency is always a hot topic when we're talking about, any of the advanced therapies. But it is, generally so product specific that it's something that may be difficult to develop, a true standard for that, would be applicable across multiple products. So it's always on our radar in terms of what we can do to support potency assays. But I would say it's not our highest priority right now. But any ideas that the group has, I'm happy to listen. So as I said, we rely on our
So,
stakeholders to help us in that one.
which is lead me to a couple of comments I have. Could you please elaborate a little bit more about the process of how people could share methods and also the process of any lab that's interested in collaborative testing?
Right.
What they should do and who should they contact and all of that. So,
Yeah, there is probably the best thing to do is to go to the the draft guidelines, because that has the information on the email address that you can, send methods to, and that will also, you could also send, if you're interested in collaborative testing, that's also a great place to send something, and then we will have that information as we move forward. I believe it's USP vaccines, but I won't guarantee it. So
Okay, no, that sounds good. Uh, Let me see. Well, I don't know if it's good or bad. We don't have that many questions. So sorry, uh, how do you see the future of QC testing for mRNA? I mean, I would like to hear it from USP perspective and also from a CDMO perspective, because I've worked for a CDMO and it is pretty challenging, to accommodate customers. So, I, just to hear your thoughts about the futuristic QC release assays for mRNA products.
I think it's an interesting question, because I do think we're still seeing a lot of analytical development in this area. You know, I do think that having multiple assays that are, well characterized, or where we have, standards to support them, is important, because I think also in this space we have to think not only about what are you using to release internally, but then, thinking about it for in-country testing. You can use next gen sequencing for identity testing, right? But is a QC lab that's doing in-country testing going to be able to support that? And so I think this is where having some flexibility makes a lot of sense.
Yeah, no, it's so that's something that we're focused in on. It's like and so I think you kind of see actually even from the slide there is that for every addiction PQA, we're trying to find at least addiction two independent methodologies. So it's actually instruments. So just like if so it's actually getting on a plane, right? There's so much so it's actually the redundancy in sensors and et cetera. And so that actually is, so that's what makes so it's actually the airlines actually so safe, right? Is because sure it's actually sensor after sensor, and they're all based upon so it's actually different technologies, and the models are all built on that. It's very similar to here as well, that you need kind of multiple attributes, or it's actually multiple methods that are independent, actually measuring the same thing, to really have the assurance or actual reassurance that the product that you're making is actually as safe as possible, potent, etc. So that's where I think that we need to be focused on the futuristic, and really being able to kind of actually condense that timeline down. So I'm very hopeful of that, but we will see.
Okay.
I
No,
think
I mean,
maybe
yeah, go ahead, Diane.
I was going to just add one more comment there, that I think, if I look at like what we've seen in like monoclonal antibody testing, for example, right, you do over time see that there does tend to be convergence around, in many cases, a single method or a single analytical, platform. Um, but I think with all the development and all the advancement in this space, we're not there yet. So, like as an example, in MABs, like charge variants, right? We did a survey a couple years ago, and it turns out that like 80% of companies not only use this basically the same technology, they use the same exact column, right? But in this space today, we're still seeing a lot of innovation. So I think it's going to take some time to get to that level of convergence.
No, I mean, I do agree with both that orthogonal methods gonna be a key here,
Mhm.
and eventually something will come out that you might not need all the orthogonal methods, but one will probably be uh, superior to all the others, or that gives you the right thing. So it's gonna take a while. I do agree with that. We do have a question. This is for you, Diane. How is USP collaborating with other pharmacopeias like AP, JP to establish global standards?
Sure. So I think we have both very formal mechanisms and lots of informal mechanisms that we work with other pharmacopoeias. One of the ways we work more formally is through the Pharmacopoeia discussion group, which is European Pharmacopoeia, Japanese Pharmacopoeia, USP. We've now added India and very recently Brazil. And so that's one way that we build that sort of regulatory convergence or harmonization. But we also have, other discussions more informally, to see like what are there, are we thinking along the same lines? Are we trying to get to the same goals, but maybe have a little bit different approach to doing that? So there's lots of informal conversations to try to build in that convergence where we can. um, so yeah, frequent conversations. And like I mentioned earlier, we also frequently have regulators from some of those other pharmacopoeias that are or regulatory bodies that are serving either on our committees or as government liaisons or sometimes as members of the committees as well.
Thank you. Well, I think this is, if no additional comment, this will conclude our webinar for today. I thank you both for your time, and I thank the audience for for your time also. So we'll finish a little bit early today, and everybody, have a great day. Thank you so much.
Thank you.
All right.
Bye.